Good afternoon. Welcome. I'm sure some of you were at the last session, but for those who weren't, my name is Stephanie. I'm one of the Science Festival volunteers. Welcome to this further session on genetics. Our speaker now is Shona Kerr, who's the project manager for the Viking Genes study. She worked closely with Professor Jim Wilson to define and implement the online Viking 2 and 3 volunteer recruitment processes and the return of the genetic results. Shona earned a PhD from King's College London and did research on DNA at the University of Oxford and the MRC Human Genetics Unit before she became a project manager at the University of Edinburgh, specializing in data protection and ethics, subjects which I suspect will be of both personal and professional interest to many here today. Before we start, forgive me if you've already heard the housekeeping bit in the last presentation. No fire drills are planned, so if you hear an alarm, please take it seriously. The exit doors are behind the screen over there, and the one at the top where you came in. The muster point is out in the car park, and re-entry is through the main building entrance. Hopefully we won't need to know that, but now we all do. Having said that, could I ask you also to put your mobiles into silent mode? But please do participate in the session. We've got the Slido reference up here, which is where you can submit questions, and I can present them to Shona at the end of her presentation. Or if you don't want to use Slido or can't get onto the page, we'll also have someone roving around with a helpful microphone. So at the appropriate time, just raise your hand and we'll get the mic to you. Having said that, I think we're all here to hear Shona. So Shona, over to you.
Well, thank you very much. Can you all hear me at the back? Yeah, great. Well, thank you to the Orkney International Science Festival for giving me the invitation and the opportunity to speak to you about my research work today. And I'm more than happy to take any questions you can come up with by any of the means that have been described to you. Okay, so great. So the aspect of the Viking Genes study that I chose to speak about today is that to do with getting genetic results back. So returning research results to our volunteer participants. And to tell you how we did that, I'm actually going to take a slight step back, and in today's talk, I'm actually going to begin by giving you a really short introduction to genetics, and it's partly to help to explain exactly which results we're giving back and which we are not, and also just to make sure that we're on the same page with respect to some of the terminology and jargon that we do have a bad tendency to use in our work. The main focus, which those of you who were at Jim's talk will have already heard the concept of actionable findings, but I've gone on the basis that I'll either repeat things Jim said, and there will potentially be people who didn't hear him, so hopefully we can keep on track. My focus, unlike Jim's, is going to be on the ethics and the permissions that we obtained and the processes that we use to actually return results to volunteers and that we are using. I'll say a little bit about some of the early outcomes we've had from returning results, and I'll finish up with some maybe perhaps slightly speculative discussion of the future of what we now increasingly call genomic medicine. It's quite possible that when my daughters are the age I am now, they won't talk about genomic medicine, it'll just be medicine. Okay, so here we go with the introduction to human genetics. So the human genome is term we use for all the entire genetic content of the human being. That turns out to be about 3 million base pairs. Now, already that's what do I mean by a base pair? A base is just a type of molecule, and in DNA, there's 4 different bases. The chemical names for which one begins with A, one with C, one with T, and one with G. And the amazing beauty of the DNA, the genetic material, the DNA molecule, is that the information coded in one strand automatically specifies the information in the second strand. So it's base pairs. There's a pairing and a double helix structure. So the DNA is not one long strand. In humans, it's packaged into 23 chromosomes. And there's no particular magic in the number 23. Some animals have more chromosomes than humans and some have fewer. We have 23. And that's where the ancestry company 23andMe takes its name. So that's a mind-boggling amount, 3 billion A's and C's and T's and G's. And I think it's quite well illustrated in this picture from the Science Museum, where someone took it upon themselves when the Human Genome Project, which was the initial project to sequence the human genome, when that was completed around the year 2000, this is actually a printout of all the A's and T's and C's and G's in volumes according to chromosome. It's a little bit pointless because it's static, unreadable paper version, but I think it really helps to illustrate the information content that we all carry. The mind-boggling size of that is slightly reduced when we think about the number of units of inheritance, which is effectively what a gene is. And in the human genome, there's thought to be about 20,000 of those, so a slightly more manageable number. The Human Genome Project stimulated a huge leap forward in the technology that we can use to analyze DNA. And there's now roughly 3 main types of technology used. And I thought I'd just put in one slide to explain those because I think it can get a little bit confusing. So if people talk about markers, which is often the case in, for example, a genealogy test, it's not the whole genome that's been sequenced. It's not all 3 billion base pairs. It's just little islands across the chromosomes, usually about 1 million or so. And these are places in the DNA that are known to vary in human populations. So they're really useful for sorting out who's related to whom, what ancestry people have. And that's called genotyping. It's relatively low resolution because it's only a tiny fraction of the genome is being surveyed, which is perfectly fine for lots of types of analyses. But these are really, most of these markers are not within genes, and so there's little or no direct health information there. But it's used a lot in research, and I'm going to give one example of that shortly. The step up from that is what we call exome sequence, and that's about 3% of the genome, and it's mainly the genes. So we're focusing in on these units of inheritance, and it's high resolution. So we're not looking at very much of the junk, the bits in between genes. Colleagues who study the junk call it dark matter and get research grants written to fund them, but I call it junk. So it does miss a few important things. I'm sort of joking. There's lots of complexity that we miss if we just look at exomes. And this is used a lot in research, including our own Viking Genes research, and also increasingly in medical genetics in the NHS. And then finally, we have whole genome sequence. There's a few different technologies underlying that, where the entirety of a person's genome is sequenced at high resolution. And this does definitely mean that a lot of junk DNA is sequenced, but it also finds important things, and increasingly that's the case going forward. At the moment, mainly it's used in research only. Now, genes are the unit of inheritance, but DNA is just this static molecule. And in order for genetics to work, the DNA has to be made into something decoded in a way. And in fact, the genetic material is a recipe book for proteins. And proteins are everything that we have in our bodies, practically. Many of the important things we have. Proteins have structural roles, for example, our hair. They're used in defense. Antibodies are protein. All sorts of transport through our bodies in the blood. For example, hemoglobin is a protein. All the signals, hormones act on proteins usually. Neurotransmitters act on proteins. And a class of protein is an enzyme, which is in effect a biological catalyst, so catalyzing all the many reactions that are going on inside our bodies all the time. And interestingly, so most medicines don't act directly on the gene, they act on the product of the gene, which is the protein. And we're not all the same. We're all very similar in the genes we carry, but we're all different. And this variation is a good thing, and most of it doesn't matter in terms of health. However, some of it does, because particularly if the variation that occurs naturally has happened within a gene, that can either have disastrous consequences or mild consequences, or still no consequences. But the DNA variation influences pretty much everything we can measure in biology and health, either by changing the protein structure or function or the amount of protein made, etc., etc. Okay, so that's the end of the introduction to genetics. A quick recap. We're looking at a very long string of A's and T's and C's and G's. When we sequence DNA, we're reading those letters off. And that DNA, in turn, is a recipe for proteins. And changes in DNA sequence that occur naturally can result in no effect at all. If they're in between genes in the junk, there can be a useful research tool, but they have no direct effect. But sometimes they have important health consequences. And these variations in the DNA sequence do contribute to all our traits. And there's a picture of the beautiful double helix of DNA. So, Viking Genes is a genetic study. To study the human genome, we need to sample people, and we need to do this ethically. So, over the years, the Viking Genes team, led by Professor Jim Wilson at the University of Edinburgh, has recruited people into these research cohorts, and particularly starting with people with ancestry from Orkney, then from Shetland, and more recently from the Hebrides. And there's a lot of information on the study website there, and more recently, the work I was involved with, we were able to expand the recruitment by making use of the wonders of online consenting and questionnaire completion. And that's why we were able to get people all across the world to send us their samples through the post. A simple saliva sample contains enough genetic material that with the sophisticated lab techniques we now have, we can extract enough DNA for really quite detailed analysis. And when the project started, it was really quite a feat to have genotype data on all these samples, and a huge amount of valuable research can be done with genotypes. Then a few samples were whole genome sequenced when the University of Edinburgh invested in a new platform technology for that. And more recently, we've been able to exome sequence, so that's sequence the genes, all 20,000 genes from about 10,000 people. So it's an absolute mass of data to analyse. I've just told you Viking Genes recruited people with ancestry from Orkney, Shetland and the Hebrides. You know where that is. we called it Viking Genes. It's a really useful tag, and Jim and the team did prove that people from Orkney and Shetland have the highest amount of Norse blood of anyone in the UK. Not really terribly surprising. And in fact, as I'm sure most of you know more than I do, the Vikings really got a long way across Europe, and there's some really fascinating intersections now between archaeology and genetics, where ancient remains can say a massive amount about the history of these people. But for us, it's just been really useful, and on our social media, it lets us put pictures up of this cute little dog with his Viking costume, etc. So, onto the science. The 4,000 people we recruited initially in Orkney and Shetland came into clinics and had a whole battery of measurements made. And one of these is the ECG, which is a measurement of the electrical activity of the heart. I'm sure many of you are very familiar with it. And in fact, that heartbeat is part of the logo for the Viking Genes study. Turns out the ECG is a really incredibly useful measurement clinically, and the trace can actually be converted into data by dividing it up into various intervals and then looking at the distribution of that data in a population. This is just one example of a part of the ECG data. And in fact, if you look in all populations, not just those in Orkney and Shetland, but everywhere, there's a lot of these kind of broadly bell-shaped curves of normal distribution. And that's true for things like height, cholesterol level, all sorts of measurements that we're quite familiar with having done in the clinic and in research. Now, these traits that vary quantitatively can have a particular type of statistical genetic analysis applied to them. And it's quite a hard thing to explain, so I've basically bottled that. I just told you it's a fancy statistical technique called genome-wide association studies, so GWAS. And the more people that have genetic data and the measure, the more powerful the research is. So the Viking Genes data has contributed to a massive worldwide effort, which has made all sorts of fantastic, important, basic research findings over the years. So everyone contributes, but there's nothing individually clinically relevant from this type of study. I'm just going to quickly give you an example. So again, I've told you that data from ECGs varies quantitatively in populations. Here's a so-called GWAS result. This is a research paper. You can see how many names are there, including Jim's. And that's because of this collaborative effort worldwide, not just people from Viking Genes, people from other studies. And what it does is it pinpoints regions of the genome close to genes which are involved in creating that variation in the measurement. And we can then gradually build up a picture of what all these proteins encoded by the genes are actually doing. And I think this is quite a nice example because nearly all of the so-called hits, the genes that were identified by this method, turn out to have a role in cardiac muscle contraction, which is just what you would expect for looking at this aspect of heart functioning. And there's some genes we're quite interested in that picture, including one that Jim mentioned called TTN. So that work all uses common genetic variants that don't have immediate clinical relevance. Increasingly, as the data has become more and more detailed, we've become more and more interested in the rare genetic variants. And some of these rare variants are clinically relevant, and some of these are actionable. And this is really the key part of the talk, because there's some genetic variants that aren't good for the gene and therefore the person carrying them, but really there's nothing can be done. But there's a subset where if we know about the change in gene function, there's treatment available and sometimes even a cure available. And so they're the ones we're really interested in. And I might call those AVs. And in all populations that have been studied, there's something around the 2.5% mark. So it's not just in Orkney or in Shetland. It's really everywhere. There's somewhere between 2 and 4% of people carry one or more of these actionable variants. I'm going to spend a little bit of time telling you about the first one we uncovered, which increases risk of a rare condition called long QT syndrome. And that's where part of the ECG trace is prolonged. And so we had that data and we were able to use it to find 5 people in Shetland who carry a variant that is very rare, but increases the length of this interval, and thus increases the risk of having long QT syndrome, which in turn increases the risk of a sudden cardiac event, a severe cardiac event. The NHS and clinical geneticists in Aberdeen, who are responsible for the populations of the Northern Isles, knew about this variant and it had been quite extensively worked on. However, because we were able to look at the detailed genetics of the people carrying this particular variant, we were able to show that they fell into 2 quite distinct pedigrees, as shown in the slide, and we could just connect them genetically. They couldn't be connected in genealogical pedigrees or social pedigrees, so they weren't known to each other as being related. And that meant that the family B wasn't on the radar of the clinical geneticists. So we were able to return results. We went to the ethics committee, we discussed it with our clinical colleagues, and we worked out a way that we could return this result to the GPs of the family B people. And we did that back in 2018. And that really got us thinking that we were going to get more and more examples of this type of thing. And then some while later, we were actually contacted by one of our volunteers, Ingrid, and it turns out she was in Family B. She'd been called in by her GP. They'd done some tests and they found she did indeed have a prolonged QT interval. She was therefore at risk of having a cardiac event. And they were able to give her medication to prevent such things. And she knows she's a very fit lady, but she knows not to, for example, run a marathon. 10K might be fine, but don't overdo it. So she's been very grateful, I think, to the study to have this knowledge before anything bad happened. And that was further motivation for us. But we're starting to get into really quite deep ethical issues here around this sort of thing. We do have a very robust system in the UK whereby all research work involving human participants has to go through quite a rigorous process, even if no drugs, it's not a clinical trial, it's just an observational study with a sample taken for DNA analysis, as we have. But it's still quite a rigorous process. So these research ethics committees are there to safeguard the rights of the and protect the volunteers. And the ones we work with are run by the NHS. So increasingly, the ethics committees are having to think about this relatively new term, genomic medicine, where it's not about treating people so much as preventing something bad happening. And I think that's quite a useful little set of words. So it's predictive because we're seeing things in the DNA. Preventative, that's a diet, which for some conditions actually is the way to prevent problems occurring. It's personalised, it's not a one-size-fits-all, it's tailored to the individual because of their genetics. And it's really vital that the people who are involved in the genomic medicine participate and feel empowered as to why they're doing so. And I'll come back to that later. So, actionable genetic variants, the things we might want to return. Again, going back to genetics, almost all of these act dominantly, which means you only need to have one copy for the effect to be seen. And they are rare, but collectively, when looking in most populations, they're somewhere around 2%. Now, when we thought we wanted to return actionable variant results, that's not the same as giving people all their genetic information. It's quite different. Oh, sure you can work that out. It slipped slightly. So if there was to be a genetic variant that predisposed to a condition for which there was no treatment, we wouldn't be mentioning that. And we're also not in the business at the moment of returning results where someone's merely a carrier and could have an affected child with another carrier, because that individual is healthy. So we don't return those. And occasionally there are non-parental events, most usually misattributed paternity, and that's not the sort of thing we want to be returning to our volunteers. But we did think it was important, we do think it's important, to try to notify them directly, not by this convoluted via the GP thing that we'd managed to work out before, if we found they carried an actionable variant. And we took this, lots of colleagues said, no, that'll be too difficult. Don't think that'll work. We took it to the Research Ethics Committee, and actually they were really very positive and helped us to find an ethical way forward, which is fantastic. And we are one of the first research projects in the UK to have been given permission to do this sort of thing. Okay, so before we could return, we had to have a plan that the Ethics Committee was happy with and that we were happy with and believed was achievable. So we had to develop a plan. Really important part of this whole thing is to have informed consent from the research participant. And it's also important that that should be optional, because not everyone wants to know, and respecting that right not to know is pretty vital. I mentioned our online study, where from the start we were able to build in this idea of returning results, and 98% of people said yes, they would. So that's very encouraging. For our clinic-based studies, this wasn't considered at the time of recruitment and consent, because such exome sequencing really was pie in the sky. It was something for the distant future. So we had to go back to that study and find people and explain what we wanted to do and why, and take new consent. So that was a really big exercise that actually is still ongoing. We had a fantastic response. Once we'd got all that sorted, we obviously had to collect and analyse the genetic data. And then from that huge volume of data, we had to consider which results to return. We wanted to be as sure as we could be of the results, try and confirm them, then get round to disclosing them, and then follow up how that went down and monitor outcomes. And in fact, this is so new that we were able to publish a paper last year explaining how we were doing this and how others might consider doing so. So this is a sort of flow diagram of how we went about achieving this. We've sort of alluded to most of the components before. We were able to extract DNA either from blood or saliva in the spit and post sample, sequence the exomes, the genes, analyze this. That's a huge step, really complicated, to narrow down into the regions and genes of interest. Do as many checks as we could, repeat the lab work using a different technology on a different sample of DNA, and then provide our clinical colleagues with this information and engage with them on which results we should be returning. And I think that's quite an important part of our process, because ultimately it's a clinical decision based on our research data. and that's Zosha Midrojska, who is our close colleague up in Aberdeen, NHS Grampian. And I started by telling you about the 3 billion base pairs and the 20,000 genes. Actually, we're only looking at 81 genes, so it's a tiny fraction of the genome. And it's not just our decision. There's lots of geneticists worldwide really working out which are the most important genes that can actually be considered to be actionable, for which there's treatment if they're found. And there's lots of challenges around this type of work. So many of the people that clinical geneticists normally see in the NHS already have a condition. They do go out into their relatives, however, and they're usually healthy. But most of our volunteers are likely to be healthy. So it's a different type of counselling and consent that's required. Can't just dump a huge, well, we could, but we wouldn't be very popular if we just suddenly dumped all our data on our people and expected the clinical geneticists to just pick up all the workloads. So we've had to kind of work with them to make sure that we're not overburdening them. And although the genetics tests are actually fairly inexpensive, if someone has, for example, a predisposition to a cardiomyopathy, that might involve quite expensive tests. So there's a cost to the NHS as well as, we believe, a benefit. Because really what we're in the business of here is preventative medicine. So even though there's an upfront cost, it could save development of disease, save lives, and save future money. Finishing off my process flow, once we've agreed which variants to return, Viking Genes, basically Jim Wilson, sends a letter which the ethics committee agreed the wording of. And basically it says, we've made this finding in our research, we've checked it carefully, it's in such and such a gene which can predispose to such and such. We recommend you contact our clinical genetics colleagues, and they're waiting to hear from you. So, quite a challenging letter to receive, but one which the ethics committee agreed, and we firmly believe is worth sending. When the participant receives that letter, they could put it in the bin. That could be the end of it, or they could take it forward. If they do choose to take it forward, they basically enter the whole NHS pathway of counselling and advice, and they get a new sample taken so that the clinical decision isn't directly based on our research findings, but they're checked to a clinical standard. And of course, we've done such hard work to get there. So far, none of them have been wrong, and they're unlikely to be wrong for a whole range of reasons, not least of which we're looking within pedigrees, so everything makes sense within the context of the information we have about these people. And then once the result is confirmed, the action can be taken to reduce disease risk, and also into family members, and that's called cascade testing. So if people within the family are also interested in being tested for the same variant, it can spread out across their pedigree. So this is our famous BRCA1 variant from Westray, which is an obvious one to return to people. And it got a massive splash in March last year. I think Paul says everyone gets 15 minutes of fame. I think that was mine. I think even the University of Edinburgh press office were a bit surprised at quite how much it just captured people's imagination. And I think it was partly the special nature of Orkney and Westray that kind of just resonated with people. So I think that was very motivating. What was particularly important, though, as well as all the BBC stuff, was that I think we've managed to take, particularly Jim has managed to take the local people along with the team, and the Arcadian had this very positive spin and explanation of what we were doing, which was very important. So again, just a recap of the process by which we're returning results. There's the kind of lab research end, and then the clinical agreement, and then finally the actual contact and the decision by the volunteer whether or not to take it forward. And we're still sending letters. We've completed all of the BRCA1 and BRCA2 letters, but there's quite a lot still to work through, particularly with our new online volunteers, because we only received the data, the exome sequence data for them quite recently. And as well as the participants themselves, the test can go out into their families by cascade. And that's exemplified in this slide by some of the outcomes. So even though the number of people in our study might be quite small, by the time you've cascaded out, I think what's the data there? Yeah, 56 people were eligible for cascade letters from just 5 consented carriers, Viking genes. So it doesn't mean all 56 of them will be carriers, but it's still quite a large number. And in all 3 of the rare founder variants we've really looked at in depth so far, we've found families that the NHS wouldn't have been able to find through the social pedigrees, through cascade testing. And we've noticed that the recipients of the letter take quite a variable length of time to return, to respond to the NHS. Some respond right away, some take a while to think it over, and some, so far, a small number, choose not to do so for whatever reason. It's a lot of words on this slide, but it's really making the point that it's not so much. So there's these founder variants in the isolated populations in the Northern Isles. All populations do have variants in these genes, urban, everywhere. It's just they're more, there's a higher number of different variants. And the overall percentage isn't that different. It's probably a bit higher, particularly in Shetland, but it's not hugely different from any other urban population. And in fact, given the better environment in the Northern Isles, people from Orkney and Shetland are probably overall in quite good shape health-wise, and some of the life expectancy data actually backs that up. So it's not that there's this plague, it's more an opportunity because of the simplification of the gene pool to home in on these few key variants and do really a lot of good for relatively little outlay. And also the second quote from our clinical colleague is that developing cancer is a really complex multi-step process, and it's not just about carrying a risk variant. So some people who carry these risk variants won't go on to develop cancer, and it's a complex mix of environmental factors with the genetics and with other genetics in other genes that the person might carry. And it's also not just cancer and heart disease variants that are on this list of 81 genes. There's also some metabolic conditions. Right at the bottom, we've got this hereditary hemochromatosis or iron overload, which is something we're quite interested in. There was even a show at the Edinburgh Festival Fringe last month from a couple of brothers who are affected by that condition. And that one's actually slightly lower in the Northern Isles than some other parts of Scotland. So it's just how the population has evolved. Well, is that true? I was going to say most research, but an awful lot of research done in universities is just for the sake of gathering knowledge. And sometimes that knowledge can turn out to be fantastically useful in unexpected ways. So I'm a great fan of that kind of blue skies research, it's sometimes called. But actually, increasingly, the funders and the important people at the university, they really like to see impact and translational outcomes from the research that's going on. That's fair enough up to a point. So one of the things that we can do now is actually to influence policy, and that's really quite unusual, I think, for geneticists working in the basic research domain. And we've actually been able to get the Westray variant added to the NHS Scotland Genomic Test Directory just a couple of months ago, which is a start, but it's by no means finishing line. So I just thought I'd finish up by saying a little bit about what's next. So as I mentioned, we're still working through the complex analysis of the exome sequences in the people who were recruited most recently. And there's lots of work to be done, partly with our clinical colleagues, to collect information about how well this has been received, how many responded, how many other carriers did this lead to, etc. And we have to continue. We couldn't do this without the support of the volunteers. So we have to communicate and explain and survey and listen to what the volunteers are telling us. And of course, as academic researchers, we want to publish papers, and we want to also tell people that perhaps they could consider in their own research studies attempting something of what we're doing. And I think in general, as I mentioned at the beginning, genomic medicine, it's got a very interesting future. Can we switch the NHS from being a more treatment-based system to a more prevention-based system? We're not alone in being interested in this concept. There's a massive UK project just getting going called Our Future Health, which aims to recruit 5 million people. I mean, that's a lot. I believe it's not going spectacularly well, but they are nonetheless busy recruiting through Boots the Chemist, actually. And I think that's quite an interesting one because it's you can see it's got an NHS logo, but there's also quite a big industry partnership behind it. And I think that'll be an interesting one to watch and to see if they do indeed manage to recruit basically 10% of the adult population of the UK. It's a big ask. I wouldn't want to project manage it. This was really interesting. So many of you will probably know that newborn babies get a little heel prick test done with the consent of the parents, a tiny little spot of blood taken, I think, usually within the first 10 days after birth. And that's tested for a very small number of ultra-rare genetic conditions for which there's treatment. And that's been accepted and ongoing for really quite some time. And increasingly, originally it was a biochemical test. Increasingly, that can be done genetically. Rather than looking at the protein, you can look at the genetic material and analyse that. And there's been talk of expanding this newborn screening programme. And there's a really interesting pilot now running in NHS England to sequence the genomes, the whole genome of 100,000 newborn babies. With the consent of the parents, but obviously not with the consent of the baby. And the idea is that it will uncover more of these incredibly rare conditions and allow them to be treated before the child develops a problem. So I think there's a lot of ethics around that. There's a lot of surveying, and there's a lot of careful work being done to see how that works out. I'm not sure I'd put my child in it if it was me. If the child was ill, I would. But all that data's out there and can't really be got back. So interesting. So that's me, really. I hope I've explained a little bit about what we're doing, why we're doing it, and given you some thoughts about some aspects of the future of medicine and where we might be at in the not too distant future. Of course, I have to thank the team. All this work is very much team science, particularly Rachel, David and Craig, all headed up by Jim. We work closely with the NHS Grampian team. Our main analyst for all this amazing amount of data is Dr. Lucia Claridge, and we've also got a great lab who process our samples back at the University of Edinburgh. And again, absolutely couldn't be doing this without volunteers. Thank you for listening.
Thank you so much, Shona. That was a really rich presentation, and I'm sure it has generated as many questions as it has already also answered many already. I have a number of questions on the Slido here, but we can also talk to real people in real person. If anyone has a question, please raise your hand. I'll try to alternate Slido and folk in the room. I'll start with one off Slido just to get us going. The first question was about how the actual research was done. At the moment, you're working with blood and spit. Could this research also be applied on gametes?
It could, but it won't be within our plans. So gametes are the sperm and the eggs, and that's a really interesting topic, but it's not one that we would be getting involved in.
I suppose it would be somebody else's grants.
Yes.
Next couple of questions are about, you alluded to the really tricky nature of sharing information, what information gets shared with whom and how. And I think it was very heartening to realize that some very sharp people are thinking very carefully about this, that it's not a one-size-fits-all approach. One of the questions was where you find conditions with no treatment at the moment. Are those shared with other researchers to help direct innovation?
That's a really good question. Indirectly, yes, because all of our data is available through the appropriate channels under contract to other researchers. And there's quite a sophisticated mechanism whereby universities interact with each other and put agreements in place, and then the data can be shared. So if another researcher had a particular interest, then that probably is something we'd be keen to do, but it wouldn't. It would be done in such a way that the identity of the individuals would not be disclosed. And when I went through my complicated protocol, of course, it would have been far easier if we could just tell our clinical colleagues, this person living at so-and-so place has such and such variant, but that's not part of the consent. We have to empower them to make the decision, the individual. So we've got this quite complicated process where we only discuss variants with our clinical colleagues. We don't discuss people. And then they also disappear from our site when they contact the clinical geneticists, because that's not our business unless they choose to make it our business. So it seems complicated, but it seems to work.
No, thanks for that explication, because I think we can well imagine, you've discovered a rare variant that can relate to some horrible but rarely seen condition. On the one hand, you don't want to just put it on the front page of the paper. On the other hand, it would be a real waste if the connection wasn't made with the researchers in that rare condition. So somehow they can at least be flagged. Look at this. So that's very encouraging. Thank you. Anyone in the room? Are people getting me out? Lady up at the back in pink. The microphone is on the way to you. Thank you for helping the volunteers get our steps in.
My husband and I can't remember if we responded to the letter or not. How could we find out whether we had responded?
So the offer of return of results, the consent.
Yes.
If you get in touch with the study, we can look up and tell you.
How do we do that?
So email email is probably the best. It's on the website, but I should have a leaflet. It's just Viking Genes at ed.act.uk. Viking.genes@ed.act.uk. Yes. Oh, thank you. Yes, that website there.
Yeah, there.
Yeah. Okay.
Yeah, we'd be happy.
So for the people who took a while to respond, it's not too late.
Absolutely, and not too late. No.
Oh, that's brilliant. Anyone else in the room? Not at the moment. A couple of questions about how to get this information known and most useful. I'll pair these two because they seem rather related. Could we start to see genealogy research services, who you mentioned, combine work like yours to provide a more in-depth service? And the other question is, can we expect this type of work to become available to the public to help the medical services become more responsive?
So take the first question. I think, genetics is the study of relatedness. So the more of that that's available to help with the analysis, the better. And in fact, some of the genealogy companies, most prominently 23andMe, have also got a health component. So I think that's an interesting development, and it may continue. Most of the studies, including Our Future Health and that I mentioned at the end, this new 5 million, and also the existing UK biobank, the 500,000, they actually asked very little about people's family structure. And a lot of that can be deduced from the genetics, but it's not really something I think that was front and centre when that study was designed. And I'm guessing it'll be true also for Our Future Health. So possibly an opportunity missed.
And do you think that would be an opportunity for, to be crude, Ancestry, 23andMe and so on, their businesses? Is this a commercial opportunity, a health service opportunity? I'm being a bit provocative.
Yeah.
I'm very curious.
So I think potentially what 23andMe have focused on is the markers, the bits that aren't usually directly health relevant. So they're more at the blue sky end of things than the individual rare actionable variants. And I think it would be a big shift for them to get their people to move in that direction, but it's not impossible.
Well, you mentioned when your daughter is your age.
Yeah.
Perhaps by then. I was reminiscing the other day that I remember seeing the film 2001, Space Odyssey, and that we all marveled at the concept of a video call. And of course, it was only available to astronauts because it was so high tech. And now, I have them every day.
Yeah. I'm still waiting for my jetpack, though.
I know we were promised jetpacks for those who like indie music. Any more questions in the room? No, you've given everyone food for thought. Well, if there aren't any more questions, I'm going to close by thanking you, Shona, for a really fascinating session. Thank you very much.
You're very welcome.